Everything that happens in your body right now, from thinking to breathing to digesting, it's all a series of chemical reactions. But what controls the speed of all that? How fast does life actually happen? Well, that's what we're going to get into as we dive into the absolutely fascinating world of enzyme kinetics. Okay, I know, that sounds like the setup to a really weird joke. But trust me, there's a single, powerful biological idea that links every single one of those things. Any ideas?
Yep. The answer is enzymes. It all comes down to controlling these incredible little engines of life. When you understand how to hit the gas or pump the brakes on them, you can do amazing things, from keeping food fresh to creating brand new medicines.
So what is an enzyme, really? Think of it like a tiny, super efficient worker on an assembly line inside your cells. It grabs a raw material, helps it transform into a finished product at lightning speed, and then, and this is key, it lets go, totally unchanged and ready for the next job. They are the catalysts that make life happen on a timescale that actually works.
Okay, so if we want to control how fast an enzyme works, what's the first dial we can turn? Well, the most obvious one is just controlling how much stuff it has to work with.
And in biochemistry, that raw material has a specific name. We call it the substrate. It's the one molecule that a particular enzyme is perfectly shaped to grab onto. Now, to make this really stick, let's use an analogy.
Let's just imagine our enzymes are a fleet of taxis, all lined up and waiting. The substrates, those are the passengers who need a ride. The chemical reaction is the trip across town, and the product is that passenger getting to where they need to go. Simple, right?
So check this out. It's just common sense. If you've only got a few passengers milling about, not many trips are going to happen. The arrival rate is low. But as more people show up, as you increase that substrate concentration, more taxis get filled, and the rate of arrivals picks up steam.
But, and this is a big but, you eventually hit a wall. A point where every single taxi is occupied. It doesn't matter if 100 more passengers show up at the stand. You can't make any more trips because there are no more taxis available. In biology, we call this saturation. The enzyme is working at its absolute top speed.
All right, so besides just the amount of raw material, what else can mess with our enzyme speed? Well, it turns out they're pretty sensitive to their surroundings, especially temperature and pH.
Temperature is a real double-edged sword. A little bit of heat is great. For every 10 degrees Celsius bump, you can actually double the reaction rate. But if you push it too far, things go south, fast. The enzyme is a protein, and it holds a very specific, intricate shape. Too much heat causes it to unfold and lose that shape. That's called denaturation. It's like melting the taxi. It's not going to be giving any more ride.
And boom, this takes us right back to our original question. Why do we boil medical tools? To denature and destroy the enzymes and antibacteria. Why do we put food in the fridge? To slow down the enzymes, then make it go bad. And that hibernating bear? It drops its body temperature to slow its whole metabolism way down, saving energy for months. It's all about temperature control.
And just like with temperature, every enzyme has a pH level where it's happiest. It's a sweet spot. For most of the enzymes humming along in your body, that's right around neutral, somewhere between a pH of 6 and 8. You go too acidic or too basic, and their activity just falls off a cliff.
So what's really going on here? This quote nails it. It's all about electrical charge. The enzyme needs to have the right positive and negative charges in the right places to properly grab onto its substrate. If you change the pH, you mess with those charges, and suddenly, it's like the taxi driver's hands are too slippery to open the door for the passenger. The connection just doesn't happen.
Okay, so we've got substrate levels, temperature, and pH all affecting the reaction speed. So how do scientists actually measure and predict all this? Well, they use a really elegant and famous mathematical model. And you know, this brilliant idea didn't just pop up overnight. It was a real team effort over decades. It started with a theory from Victor Henry, then two incredible scientists, Leonor Michaelis and Maud Metten, came along and proved it with experiments. And later, it got refined even further. That's just beautiful science in action.
And this is what they came up with. Now, don't get scared by the letters and brackets. You absolutely do not need to memorize this. The big idea here is to think of this equation as the dashboard on a car. It gives us a simple, at-a-glance summary of how well our enzyme is performing.
And on that dashboard, there are two numbers that matter most, Vmax and Km. Vmax is the easy one. That's the speedometer redlining. It's the absolute top speed, when every single taxi is full and driving as fast as it can. Km,
Km is more interesting. It kind of tells you the enzyme's personality. It's the amount of substrate, the number of passengers you need at the stand to get the enzyme working at just half of its top speed. A low Km means the enzyme is really grabby. It has a high affinity. It doesn't need many passengers around to get going. This is crucial for enzymes that have to respond to tiny amounts of something, like a hormone. They need to be super sensitive, and that means a low Km.
All right, so we've talked about what sets the speed limit and how to measure it. But what if we need to deliberately slow an enzyme down? What if we need to put on the brakes? Well, that's where inhibitors come in. And this is absolutely critical for modern medicine.
An inhibitor is exactly what it sounds like. It's a molecule that gums up the works, binding to an enzyme and stopping it from doing its job. And this is a huge deal, because so many of our most important drugs, from common painkillers to powerful antibiotics, are designed to be enzyme inhibitors.
So how does it work? Well, one of the cleverest ways is called competitive inhibition. You design a decoy molecule, the inhibitor, that looks almost exactly like the real substrate. It's so similar that it can fit perfectly into the enzyme's active site. But once it's in there, it does nothing. It just sits there, physically blocking the real substrate from getting in. It's basically like someone getting into the taxi, locking the doors, and then refusing to tell the driver where to go. The whole system grinds to a halt.
Okay, let's pull back for a second and bring it all home. We've talked about taxis, temperature, decoys, but why does this all matter so much? Why do we care about the speed of these tiny engines? Well, because understanding this speed is fundamental to understanding life itself. It explains how our entire metabolism is regulated, it gives us the power to predict how cells will react to different conditions, and maybe most importantly, it gives us the blueprint we need to design smart drugs. Drugs that can go in and shut down one specific faulty enzyme without causing chaos elsewhere in the body. It's how we understand ourselves at the deepest molecular level. You know, this quote just says it all, perfectly. The whole field boils down to two things: explaining how these things work and predicting what they'll do inside a living, breathing creature. It connects the blueprint to the building.
So, we've learned to clock the speed of life's most essential machines. We can measure it, predict it, and even control it. And that just leaves us with one huge, exciting question. Now that we really understand the speedometer, what's the next part of life we can engineer, fix, or optimize? What comes next?